my name is Pinja Näkki and I come from the Finnish Environment Institute. My topic will be the fate of microplastics in the Baltic Sea sediment, which is also the topic of my PhD work. And I just started my second year this January, and therefore I will tell you a bit from my current results. But first, let's start from the basics. So what are microplastics? I'm pretty sure that you have all heard something about them before, but just to tell you and remind you that they are small plastic fragments, less than 5 millimeters by their size. And they can come in all kind of shapes, colors and textures, as you can see from the photo. And why you should care about microplastics? There is a small list, and one of the reasons is that they are distributed worldwide. You can find them from seafloor sediments, you can find them from water columns, water surface, beaches, sea ice, basically everywhere in the marine environment, and nowadays also in the terrestrial environment. Because of their distribution and small size, they can be ingested by various animals. They are able to enter food webs, and that has been checked both in the laboratory experiments and also experiments in the nature. And because of they can be eaten, they can also be transferred into the food webs and to the higher trophic levels. When ingested, these small plastic fragments, they might cause some mechanical harm to the animals. They might cause some abrasions, for example, in the digestive tract, but also some chemical harm. These plastic fragments or plastic litter, they carry a chemical cocktail in or on their surface, and this chemical cocktail is can consist of some harmful chemicals that are added to the plastic when manufacturing. For example, additives like phthalates here that can leach out from the plastic particle when it's in the environment or inside an animal. Moreover, some harmful contaminants from the environment, like PCBs or DDTs, can also adsorb to the plastic surface and therefore increase its harmfulness to the marine life. And finally, they are also impossible to remove once they have entered the environment. They are so small that you can't pick them all, so they will stay there quite a long time, especially when you think that plastic is really persistent material. It can take decades before it decomposes. And finally, I would like you to look at this figure. It's about global plastic production, and it shows the production that has started from 1950s and grown since that. And the graph shows the production in million tons. We have now exceeded 300 million tons per year, and as you can see that the graph is going quite high there. That's the estimate of how much plastic will be produced in the next decades. So this is a cumulative problem, and I hope that now I have convinced you that this is important. But why study seafloors? Well, as I said, many microplastics have already been found from the seafloors, from shallow areas to deep sea floors. And actually, it has been estimated that most of the microplastics will eventually end up to the seafloor. In soft bottoms that can be colonized by animals, the animals alter the sediment structure in a process called bioturbation. And this bioturbation transfers solutes and particles in the water-sediment interface. And I wanted to know if it also could affect the distribution of plastics on the seafloor. My project is about the fate and impacts of microplastics on the seafloor, and I view it as a puzzle. And the first piece of the puzzle was to solve what happens when the plastics sink to the seafloor. So the first step was to study whether bioturbation could actually transport microplastics deeper in the sediment when they have just been sunk to the sea bottom. This experiment was done here in Tvärminne, and it started by generating a large amount of microplastic particles. And I wanted to use fishing line to make these particles because fishing line is one of the most common trash items that you can find from the sea for obvious reasons. And I cut the fishing line with this kind of tissue chopper. It's a device that is usually used for cutting or slicing mice and rat brains, but it's really suitable for also this kind of work. It took me 2 weeks, and it was maybe the worst 2 weeks of my life, and also maybe the worst 2 weeks of my lab mates, because this device is keeping quite a lot noise when cutting these fishing line pieces. But anyway, I cut 3 different sized pieces that were all different colors to study if the size would have any effect on their transportation. And this is my experimental setup. So I had control units filled with sediment, not any animals in there, and then animal units with common benthic invertebrates from the northern Baltic Sea with animals. And you can see some bioturbation has been happening. And I used all of these animals together to monitor kind of the community effects on microplastic transportation. And this experiment lasted 3 weeks, and after 3 weeks I sliced the sediment according to depth into 6 different layers. This graph shows that in y-axis there are the percentage of found microplastics in control and animal units, and the different colors in the bars represent the different layers that were cut from these sediment units. And as you can see, when you look or compare the animal unit to the control unit, that in the animal unit, the first layer contained less microplastics than the control unit, and that means that the microplastics had been transported deeper in the sediment. And you can also see them in the second and third layers, which contained more microplastics in animal units compared to the control ones, and these differences were significant and indicate that the bioturbation by these animals could transfer microplastics until the depth of approximately 5 centimeters in the sediment. In this study, I also checked if the animals ingested microplastics, and these results are coming next. So the Baltic clam, Macoma baltica or Limecola baltica, actually ingested microplastics. 25% of all the clams used in the experiment had eaten microplastics, which is quite high percentage. But the particle amounts inside each clam was quite small, ranging from 1 to 15 particles per clam. However, the other animals used in the study did not ingest anything, and that is because the microplastics were too large when we think of how small or large pieces these animals could actually ingest. So the plastics were too big for them. And the output of this study, which is now published, one of the outputs is the recommendations that we can now give for microplastics monitoring in the Baltic Sea. There is a lot of concern on what kind of concentrations we could find from the Baltic Sea, and many countries would like to take samples from the sediments. And so far, many people have taken the sediment samples but used only the 1 or 2 first centimeters of the sediment cores for these microplastic analyses. So according to our results, we would recommend using a thicker layer to really get a more reliable picture of the true abundances of microplastics on the seafloor. At least in these latitudes, we would suggest that 5 centimeters would be the minimum. But of course, the depth where these microplastics may end up depends on the species community and species traits, so it may be different in different sea areas. So that was the first puzzle or first piece of my PhD puzzle, and the next one was obviously to study this the other way around. So when the plastics are already in the sediments, somewhere buried, will the animals also be able to transport them back up? And this time I didn't use fishing line, I was traumatized for cutting it. So this time I wanted to use Lego bricks. So I grinded them with the kitchen grater, and I wanted to put them into distinct depth of these sediment cores, and therefore I made these delicious looking mud cakes sprinkled with microplastics in a freezer, and then put them into the right depth with the fresh sediment. And this experiment was running a bit longer, 10 weeks this time, and I used the same animals. You can here see a detailed description of the setup. So I had the same animals, same kind of cores, but this time I put them into approximately depth of, or the microplastics were put in the depth of 5 and 2 centimeters in the sediment. And I used 2 different sizes and 4 different colors, and I put them in separate layers to really see how much they were moving. The percentage of moved particles was quite small, so we could say that it's not that important process in transporting microplastics up. And what comes to the ingestion of plastic particles, only 1 clam out of 112 had eaten 1 plastic particle, so the ingestion was also much less compared to the other study. And this study helps us to understand where the microplastics end up and how they will, or what kind of animals will be exposed to them. Because now it seems that bioturbation is not bringing them up, so that the the seafloors could actually act as microplastic sinks, and most of the plastics could be more or less permanently buried in the sediments, and that might also inhibit the microplastics from entering the food chains. If we think about the animals who are feeding on the sediment surface, like many of our animals are. So these 2 studies were about bioturbation, and the third piece was started last summer, and it's about absorption of environmental contaminants or plastic particles. So if we now have seen that bioturbation transports plastic downwards but not so much upwards, then the logical question is what will happen in the sediments? Is there something that could be harmful when they are staying there? So if you can recall from my first slides, I told you that plastic is able to absorb all kinds of chemicals. So we wanted to know if this also happens in the Baltic Sea. So we started a small study about contaminated sediments. We collected sediment from 3 different spots around Helsinki area that were potentially containing harmful substances. So the areas were Rövargrundet underwater landfill area, West Harbour, and Vanhan Kaupungin Lahti Bay, which historically have been containing quite a lot of harmful substances. And we used 4 different types of plastic particles, some of them traditional, and some of them biodegradable. And we wanted to see if there is any differences in how these harmful substances may absorb these different plastics. And at the same time, we also wanted to study the microbial biofilms that will be growing on these different particles, and if there would be any degradation, for example, in the case of biodegradable plastics. But unfortunately, I have to say that this is not ready. We have the samples now in the freezer, but we haven't analyzed anything yet. So I have to say that this is still a big question mark. And what comes to my PhD work, I hope that the last piece of the puzzle would be assessing the physiological risks or effects of microplastic ingestion in these benthic invertebrates, but that's still to be planned. So stay tuned, there is more to come.